Hi, in this lecture, we're going to look more at the microscopic anatomy of the heart by looking at the cellular level. Now, the heart is different because it has your contractile cells, which are the cardiac muscle cells known as the cardiac myocytes, but it also has cells called pacemaker cells. And the pacemaker cells can depolarize all on their own. They're called autorhythmic cells. They're highly excitable cells. We will talk about both types of cells, and then we'll wrap up by comparing and contrasting the cardiac muscle cells with skeletal muscle cells and the pacemaker cells. And we'll be looking at both the anatomy and the physiology of these cells. So, as I mentioned, there's two types of cells in the heart. Now, the smallest number are the autorhythmic cells. They're less than 1% of the entire heart, and they're found in very specific areas of the heart, and these areas all have names, and you'll learn those when you learn the conducting system in another lecture. But they're excitable cells, and they depolarize all on their own. They don't need a nerve depolarizing them. Just on their own, they depolarize and cause that heartbeat to be able to occur. Whereas the muscle cells, the contractile cells, cardiac muscle or myocytes, these are the cardiac muscle cells that contract, right? They contract to propel the blood to generate pressure. So if we look at the muscle cells, contractile cells, some of this stuff you already know. You know that cardiac muscle is striated. You probably remember that it's smaller than skeletal muscle and it looks different. Skeletal muscle is long and cylinder shaped. Cardiac muscle is kind of short, fat, and branched. Now, cardiac muscle usually has one nucleus, maybe two. You might remember that skeletal was multinucleate, and cardiac muscle has a lot more mitochondria. In fact, 50 to 60% of the cell volume is mitochondria and lots of myoglobin. So that should tell you that the heart uses and relies on oxygen. The cardiac muscle cells are held together with things called desmosomes. These are a type of junction you learned in A&P 1. It's a pretty strong junction that allows the cells to kind of pull a little bit without tearing. And then you also have these intercalated discs. And these intercalated discs look kind of like lines. They're between adjacent cells, and they're abundant in the contractile cells, and we'll also see them in the pacemaker cells. But you might remember they were gap junctions. So they allow for the rapid flow of ions from one cell to the next, which is really important because the whole heart works as what's called a functional syncytium. So what that means is that when the heart is going to depolarize and contract, it does it as a unit. The atria will contract and relax together. The ventricles will contract and relax together. When we look at these cells, they look very similar as far as you have your T-tubule, you do have some SR, not as good as skeletal muscle. You might remember that the SR stored, do you remember what? Calcium, good, so it stores calcium. Muscle, skeletal muscle has a lot better developed SR, so cardiac muscle is more dependent on blood calcium. It has sarcomeres, it has actin-myosin, similar structure of the sarcomere, but the heart relies on aerobic metabolism. It can work anaerobically for short periods of time when necessary, but it's not good because remember, when you work anaerobically, you generate lactic acid or lactate, and when you get that acid, those hydrogen ions actually interfere with the movement of calcium and the action potential. So it actually is inhibitory on the heart, and it decreases its ability to form an action potential and actually contract. And so this results in portions of the heart not contracting normally, and that's called an MI, or a myocardial infarction. Now, the good news is that cardiac muscle can burn anything for energy. It'll burn fat and carb and protein. It can burn lactic acid, ketones. It burns a lot. So it's very adaptive at fuel source. The problem with a blood clot in like a coronary vessel isn't that the muscle will starve, it's that it doesn't get oxygen. So anytime we have a clot in one of those coronary arteries, it's decreasing the ability to get oxygen to the myocardium. And then that is what causes the problems. If we look at the physiology, hopefully you remember your skeletal muscle physiology. When we talked about skeletal muscle, we had our little graph, and we talked about how you had a resting membrane potential, and then you might remember that we had some kind of threshold stimulus, and it opened sodium channels and sodium rushed in, so we depolarized, and then we repolarized, and we reset. So it sound a little bit familiar to you? Well, it's similar and it's not. So we look at the contractile cells. They get their action potential from the conducting system cells. Remember, the conducting system cells are those pacemaker cells, the one that I said make up less than 1% of the heart, and they're in very specific areas. So those cells are critical because they depolarize, and they send that depolarization to the cardiac muscle. You might remember skeletal muscle got it from a motor neuron. So when the cardiac muscle contracts, it does require sodium, so it does require stimulus, and it opens voltage-gated sodium channels. Sodium enters, and it causes that depolarization to occur. Now, what happens is we're also going to, remember we talked about repolarize. Well, cardiac muscle still repolarizes when potassium channels open. And remember, we kind of looked at this, and potassium leaves. That's going to help repolarize. However, the difference is what you see here for number 2. When cardiac muscle depolarizes, when sodium enters, when it reaches the magic number plus 30, those sodium channels close. Well, what happens is when those sodium channels close, calcium channels open, and calcium enters the cell. So what we have is positive ions coming in. Now, potassium is going to try to leave. It's going to start to leave. But with the calcium coming in, what happens is you see this thing like this. And I have a better picture coming up. It causes this thing called a plateau. So the calcium enters, and it causes a plateau. And what that does, if we look at this picture, here's your cardiac muscle. So number 1, sodium channels open, sodium rushes in. Here's skeletal muscle, okay? Notice skeletal muscle was really fast. There's the depolarization and the repolarization. Now, if we come over here, though, the second step is calcium enters. That's what's going to cause this nice long plateau. That's the absolute refractory period. Remember, the absolute refractory period is the time that you cannot generate another action potential, no matter how strong. Look at it in skeletal. See how small it is? Remember skeletal muscle, we could hit faster and faster and faster and get wave summation and tetany? Well, that's pretty important if you're trying to lift a heavy weight. You really, though, don't want your heart to go in tetany. We want to have a nice, long, absolute refractory period because we want to make sure we have a nice, long, slow contraction in our heart. The heart contraction is much slower than skeletal muscle. And that's what the importance of this plateau is. So the calcium enters for the plateau, which gives us a nice slow contraction. Remember, we have to have time between contraction and relaxation so the blood can flow in those little cusps in the aortic valve, and we call it runoff into your coronary vessels. So then step 3 is then you finally open up the potassium channels fully. The calcium is going to kind of slow down and coming in, and we're going to repolarize with potassium leaving. And so we still have our repolarization, it's just a little bit delayed. So it has some similarities and some differences. Now, calcium, we knew calcium was critical for skeletal muscle. It's critical for 2 points in cardiac muscle cells. Number 1, we need it for the plateau. Remember, the plateau provides that long absolute refractory period to prevent tetany. We never want the heart to have a single sustained contraction. Calcium is necessary for all muscle to bind to troponin. Remember that it weakens the TT complex. I called that the cellular chastity belt, and it moves it out of the way so the myosin can grab the actin. So you still need it for that. So we're just going to compare cardiac to skeletal. There's a couple differences. I kind of touched on them. So number 1, the all or none law. Remember, in cardiac muscle, it's at the unit, the organ level. The atria contract and relax as a unit, and so do the ventricles. When we're looking at skeletal muscle, it was at the motor unit level. Remember, the motor unit was one motor neuron and all the fibers that it innervates. So it's a different level. The means of stimulation, we have autorhythmicity. So what that means is that the heart stimulates itself. The pacemaker cells are going to stimulate the cardiac muscle cells. That's where that action potential comes from. Where, remember, skeletal muscle, the action potential came from the nervous system. And then number 3, like we said, the absolute refractory period is much longer. You can see down here cardiac versus skeletal. See the blue? That's the absolute refractory period. And again, we don't want our heart to tetanize. Now, if we look at the pacemaker cells, the autorhythmic cells, they're going to be a little bit different. And if we look at these, they're only located in certain areas of the heart. And you can kind of see the yellow here. So they're found in something called the conduction system. The conduction system has all the pacemaker cells. And what this system is kind of think of it like electrical wires in your heart. It's not really an electrical wire, but it works like that. Because this is the system, these cells can depolarize all by themselves. And when they depolarize, they then send that action potential into the cardiac muscle. So these cells are also attached with the intercalated disc, but they work a little bit differently. This is what they look like if we're looking at their action potential. So they're very excitable. As I said, they depolarize on their own. They're constantly moving, kind of drifting towards threshold. They have gap junctions as well. However, the biggest difference is that they depolarize when calcium enters, not sodium. So the pacemaker cells are also dependent on calcium because that's what depolarizes them. That's what starts the action potential. Now they still repolarize, reset, when potassium leaves. So that part is similar. So I find it helpful to try to compare and contrast these and set up a table. So I'm not really sure if I'll be able to, I'll try to type and hopefully this will work because it'll be neater than me writing. So what I would like you to do is pause this video, pause the Edpuzzle, and see what you can fill in on your own. Take a few minutes. You got to try it first, right? Then fill in everything you can, then come back and hit play. Okay, so I'm assuming you started to fill out your table. And if we just look at anatomy, we know that they're short, fat, and branched. Excuse me. We know that cardiac muscle is striated. We know that it has lots of myoglobin and mitochondria. We know that we've got the myosin, actin, T tubules, right, sarcomere. Oh. We know that, let's just do this. Oh, maybe not. We know that skeletal muscle is not short, fat, and branched. Right? We would say that it's like long and cylinder shaped. We know that skeletal muscle can have myoglobin and mitochondria, but remember that depends on the type of muscle fiber, but it's got all that. And we also know that it has a well-developed SR, where we know that cardiac muscle, we'll just say less developed SR. So that's kind of the overview. The pacemaker cells, I don't get into too much on their anatomy. In fact, they don't really say a whole lot. They're not muscle. They're more like a neuron. So it's not muscle. We'll just say that. That way you don't have myosin, actin, none of that stuff. It's a special electrical tissue. And they tend to be a little elongated cells. Now, stimulus, right? What depolarizes cardiac muscle? The pacemaker cells. What depolarizes skeletal muscle? Remember, we need a nerve impulse from the central nervous system, and it got there through a motor neuron. These, self. On their own, they depolarize. They open up the channels and it comes in. Okay, now, the control. What controls your cardiac muscle if we're thinking about like control mechanisms? Well, one thing about it is going to be your autonomic nervous system. So we'll be looking at the sympathetic and the parasympathetic soon when we look at that. And when we talked about muscle, we always said it was involuntary because it's under control of your sympathetic. You might remember that skeletal muscle was voluntary and the division was called somatic. And the pacemaker cells control themselves through their own pacemaker potentials. Depolarization, cardiac muscle, is going to be the, we'll say this, the opening of the sodium, or let's say this way, of the voltage gated sodium channels. And remember, I guess we'll just put it all here. Remember, sodium leaves. Let me try that one more time. I was just seeing if you were paying attention. Sodium enters, and then that depolarizes, right? It changes the charge and flips it more positive. Same for muscle. How about pacemaker? What did you put for that? Hopefully you said calcium, right? Not sodium. So it's a little bit different. Repolarizing. I'm just going to copy this and fix it when I paste it. So what's going to happen to repolarize? We're going to open the voltage-gated potassium channels. Now, potassium doesn't enter. Remember, that ion leaves, and that's why it becomes more negative, because you're losing the positive charges. And they all are going to do that. Refractory period, right? We have a very long absolute refractory period. We say cannot tetanize because we don't want that to happen. If we look at skeletal, we will just say, oops, very short. I'm having some issues here. Very short. Can, right? Because we need it. We need to generate more strength. This I would just say Na because it's not muscle. Okay, the role of calcium. Looks like my table got moved a little. Role of calcium, right? So for cardiac muscle, it has two functions. It removes the T complex, right, for muscle contraction. So we'll say so cross bridges can form. Remember, that's how you can get the contraction. And then number two, calcium enters for the plateau. For skeletal muscle does not have a plateau stage, so we are just looking at it being able to move the TT complex. And if we are at pacemaker cells, remember calcium is necessary to depolarize it. So when we're looking at the heart, it's pretty important. We need calcium for our pacemaker cells. We need it for our cardiac muscle cells to contract. And we need the calcium in there so that it can set up that whole plateau stage as well. So I think it would be good to be able to compare and contrast them. So this concludes our lecture on the cellular anatomy.